The 2D-DIGE method was basically aimed to overcome the gel to gel variability, which was observed in the two-dimensional electrophoresis, and it allowed the multiplexing samples. This method was first reported by Unlu et al. In 1997, and this method finally became very user friendly, and now thousands of laboratories worldwide are using this technology to overcome the gel to gel variations encountered by traditional or conventional two-dimensional electrophoresis. So, this method, the DIGE method, involves protein labeling and separation techniques. The optical detection of the proteins is done by using the fluorescent tags, and this method performs the linear detection in the wide range of the protein abundance. So, overall one can expect a very sensitive method for giving rise to very large dynamic range of the protein detection by using fluorescent tags, and this method is giving the multiplexing and separating proteins on one gel. So, it is expected that it will avoid lot of difficulties which are encountered in the traditional 2D gel. So, let us talk briefly about a basic principle involved in DIGE technology. So, we have to discuss how to do the protein labeling, then what is the need for doing the internal standards. So, let us talk step by step. As we discussed, this is a commonly used fluorescent protein labeling reagent for the DIGE. The N-hydroxysuccinamide or NHS ester derivatives of cyanine dyes such as Cy2, Cy3 and Cy5 are used in this method. The Cy3 and Cy5 are used to label your control and your treatments. One should always try to do the dye swapping, so that there is no dye bias. So, if in one sample you are adding the Cy3 in control and Cy5 in treatment, in other sample you should do the vice versa. So, the Cy2 remains consistent for doing the internal standard. Now, for Cy3 and Cy5, these dyes, the size and charge is matched, so that the label samples can co-migrate within the gel. Now, I will talk about the labeling methods used in DIGE technology. So, when we are talking about two-dimensional electrophoresis workflow in the last class, we had looked at different type of staining methods currently available, such as Kumasi, silver staining, Cypro ruby, and in that context I briefly described about cyanine dyes. So, now in this technology, the DIGE technology, those cyanine dyes are used. There are three different Cy dyes, as I mentioned, the Cy3, Cy5 and Cy2. The excitation and emission wavelength of these three are defined. The Cy3 excitation wavelength is at 540 nanometers, emission wavelength at 590 nanometers. The Cy5 excitation at 620 nanometers and emission at 680 nanometers. The Cy2 excitation at 480 nanometers and emission at 530 nanometers. So, these dyes are photostable, they are pH insensitive and spectrally distinct. By using these dyes, the DIGE method can be used. Now, let us talk about different type of labeling methods. In DIGE, there are two different type of labeling methods which are possible. One is most commonly used, the lysine labeling, or other is cysteine labeling. Lysine labeling method, also known as the minimum labeling method, where the charge and size of dyes are matched, there are no multiple labels, and it is known as minimal labeling because it is only labeling the 3% of all the proteins. I will give you the reason why that is we are aiming for very small amount of proteome to be labeled, and that will be covered in the next slide. The saturation labeling is done by using cysteine tagging. So, amino acid cysteine is targeted to label. This is charge neutral and size matched. Multiple labels are used. It is very sensitive and used only for the Psi 3 and Psi 5 labeling. So, saturation labeling is not so widely used, but this is another method one can use for labeling the proteins, and it can be used in DIGE methodology. So, let us talk about the popular method of targeting lysine. So, why to label only lysine amino acid? Lysine is preferred amino acid, as I have shown you in this graph here, the distribution of amino acids across the proteins and their percentage distribution. So, lysine is preferred amino acid to label. Its frequency is greater than 5%. So, statistically almost every protein has at least one lysine residue. So, there is a good probability that your protein will have lysine residue and it will get labeled if you are targeting the lysine. But if that is not the case, one can always try the saturation labeling of cysteines. Now, let us talk in more detail about the minimal labeling or lysine labeling. So, Psi dyes, they react with the primary amino group of target proteins, and the N-terminal alpha amino group and the lysine epsilon amino groups, they are labeled by the process of nucleophilic substitution. So, all the available lysine labeling would create a very hydrophobic environment of the proteins. That was the case observed by the original paper derived by Unlu et al. So, to avoid that hydrophobic protein atmosphere, it is recommended to do the minimal labeling of only 3% of the whole proteome. So, that overall it is not very hydrophobic. Now, in this slide I will describe you the labeling chemistry. So, as you can see in the structure here, the structure of Psi difluor comprised of a positive charged Psi difluorophore, a linker, and NHS ester reactive group. So, at pH 8.5, the NHS ester couples with amino group of lysine in the proteins and it forms a covalent bond. The amino group of lysine residues, they are positively charged at neutral or acidic isoelectric points. Dye is also positively charged. So, the net pi will be unchanged. Therefore, the Psi 3 and Psi 5, these are not going to alter any mass as well as charge overall, and they are very well spectrally resolved. So, people prefer doing lysine labeling. It is not going to alter overall pi values. It is not going to alter overall mass, because both Psi 3 and Psi 5 will carry the same mass of these labels, and there is a good probability that your protein will have lysine residues and it will get labeled because of the cyanine dyes. So, how to prepare the sample to perform the DIGE experiment? First of all, you need to reconstitute your dye in dimethyl formamide or DMF. The lysis buffer, that is in which your protein sample is going to be dissolved, consists of 30 millimolar tris, 7 molar urea, 2 molar of thiourea, 4% of chaps, and you have to adjust the pH at 8.5. Please make sure there is no IPG buffer or carrier ampholyte, and there is no reductant added in this lysis buffer. Since you want to perform the minimal labeling, you need to add only 400 picomole of Psi dyes in a small amount of protein, 50 microgram of proteins, and during that process the reaction should be performed on ice water for half an hour. If you go back and remember about our previous discussion, to visualize a gel with the Kumasi stain, you need more protein. Even from silver and other stains, you need good amount of protein, but these fluorescence dyes are very sensitive. So, all you are looking for a very small amount of protein, 50 microgram protein quantity is sufficient for performing this experiment. Now, if this reaction of half an hour is done, after that reaction should be quenched with excess of primary amine, the free lysine, by adding 1 microliter of 10 millimolar of lysine. So, then reaction will be stopped and you can proceed with the next step. So, while you are doing this reconstitution of dyes and labeling your protein samples with the Psi 3 and Psi 5 dyes, now let us think about our Psi 2 dye and the internal standard. So, I am taking a very simple case here, where you have 4 healthy controls and 4 patients suffering from a disease. So, to represent all the proteins which are present in each of these samples in healthy individuals and diseased patients, first you need to make an internal pool which is representative of all of these 8 samples. So, you can take 25 microliter from each of these 8 samples and make it to 200 microliter of one pooled sample, which is your internal pooled standard. So, this pooled standard preparation can be performed by combining the aliquots of equal amount of protein from each samples in the given experiment. Now, the Psi 2 dye you are going to label with this pooled sample, so that can be used as a internal standard. This internal standard has many advantages, and this is actually one of the major success that how 2D limitations have been overcome. Since every protein is present in the internal pool population, so that is going to be appearing on your gel. So, there is a reference point for each gel already present on your gel, whether it is from any patient or any controls. So, the gel to gel variation will be reduced. Now, gel to gel matching and comparison will be much efficient and easier. Now, this internal standard is creating a reference point for every protein on each gel in the experiment. Later on, when software performs some data analysis, this results into accurate spot statistics and more meaningful biological data can be interpreted from this analysis. So, now we have talked about the DIGE principle, how to label the samples with different Psi dyes, why there is a need for making the internal standard, and how to label the internal standard with the Psi 2 dyes. So, let us go to the step by step procedure of DIGE method. So, very first thing is you have extracted your protein samples and you need to make the internal standards. Now, your control sample, treatment sample and internal standards should be labeled with different cyanine dyes. Internal standard is always fixed with Psi 2 dyes, but your control and treatment should be dye swapped. So, sometime it should be Psi 3 and then few samples should be Psi 5 and the vice versa. Once labeling is done, then sample should be mixed. So, all the 3 samples Psi 3, Psi 5 and Psi 2, all 3 labeled sample should be mixed in one combined tube. After that process is done, you can take that sample and do the rehydration. We have talked about rehydration step and the need for it in the previous lecture in the 2D workflow. Now, this rehydrated strip can be focused in an isoelectric focusing instrument. It is Good idea to avoid any light exposure during the rehydration, dye reconstitution process as well as during the isoelectric focusing run is in progress. Once IF is done, again you have to do the equilibration, first equilibration and second equilibration followed by SDS page. So, overall step as you can see, it is exactly same what one is doing in the two dimensional electrophoresis. Only variation here is that all your control and treatment samples have been mixed together and now they are separated on one IPG strip. Now, this strip is placed on one SDS page gel and all the proteins will be separated in one gel itself, whether it is coming from the control or treatments.
The pH of the sample will be adjusted to 8.5 using 100 millimolar NaOH. Take out the side dye tubes which are stored in a dark container. Thaw these dyes using mini centrifuge. Now label the tubes properly. 50 microgram of each sample will be separately labeled with 400 picomolar of side dyes. Add 1 microliter of side dye in each tube. Add 1 microliter of each side dye in their respective tubes. Here you can see all the 3 side dyes in different tubes. After addition of side dyes, sample will be incubated on ice for 30 minutes in the dark. Labeling reaction will be stopped by addition of 10 millimolar lysine followed by incubation on ice for additional 10 minutes. Dye gel scanning. 2D-DIGE gels will be scanned using Typhoon variable mode imager at a 100 micromolar resolution employing suitable excitation emission wavelengths. Switch on the Typhoon scanner. Place the gel plates on the tray provided as shown and insert it in Typhoon scanner. Click on the Typhoon FLA 9500 icon. Click on 2D-DIGE. You can see a DIGE window open. Before starting the scanning, name the file properly. Set area as DIGE Attendant. Resolution should be 100 micromolars. Set standard at site 2. Set the wavelength for each of the side eye. Excitation wavelength for site 3 is 523 nanometers, site 5 is 633 nanometers and site 2 is 488 nanometers. Now by clicking on Start Scan button, we can initiate the scanning process. The scanning will initiate with site 2, proceed with site 3 and will end up with site 5. You can adjust the contrast and see the gels. After scanning, gel images will be cropped properly using Image Quant software version 5.0 prior to importing the Decider 2D software version 7.0 for comparative analysis for relative protein quantification across disease and control samples.